Hi everyone, welcome to this live stream. My name is Roy van der Meel and I'm an assistant professor at the Eindhoven University of Technology in the research group of Willem Mulder. And our research focuses on developing nano-immunotherapeutics. So we use nanotechnology to make immunotherapies. And I want to thank the Kazerne, first of all, and Eindhoven Innovation Café for giving me the chance to talk about RNA vaccines for the next half an hour or so. This is of course very relevant in this day and age with the current pandemic, and I will hopefully show you in the next 30 minutes how RNA vaccines can go from code to cure. So when we talk about vaccination, we have to go back a couple of hundred years, all the way to the beginning, which you see on this slide here. And on the left, you see a cow, but next to the cow is a guy who's called Edward Jenner. And Edward Jenner was an English physician who pioneered vaccination. And the story goes that in his village he saw that milkmaids, so the maids who were milking the cows, who caught cowpox, a relatively benign disease in cows that causes pox on the others. They were not susceptible of catching smallpox, and the smallpox is a much more debilitating disease in humans. So what he started doing is inoculating fluids from these cowpox into children and people, and saw that they were protected against the smallpox virus. And this was sort of the start of vaccination. And of course now vaccination can be seen as one of the biggest medical and scientific achievements, banning a lot of very severe diseases, such as polio or the mumps, measles. Most of us, or nearly all of us, have been vaccinated as a child. And therefore we are protected against these viral infections. So before we go a little bit deeper into what the coronavirus does and how we can, let's say, fight this with these new types of vaccines, we have to go a little bit back and the briefest of information about the flow of genetic information in humans. If we take a human cell, and I made a schematic of a human cell, obviously our cells are not square, but bear with me here. Our human cell has an important part called the nucleus, which is the vault where they keep all the most important things. And of course, one of the most important things that we have is our genetic code in DNA, which you can compare to your computer's flash drive storage. So let's say your hard drive, right? If you want to read this genetic code, it is first transcribed into a working memory, just like your computer, something that's easily accessible. And you see that the working memory, which is called RNA in our bodies, is outside the vault and inside the cell. So this DNA is super stable, but it's also very much protected, and therefore it's not so easily accessible. The RNA is less stable, but much easier to read. And of course, the RNA provides a template for protein production. And the proteins in our body, which you can compare to your program or your apps, basically regulate most of the processes in your body. So this flow of information from DNA to RNA to protein is very important for understanding, how do viruses work? How do these new types of vaccines work to combat them? So with this in mind, let's have a look at a coronavirus. And before we go on, I just want to mention in the bottom of the screen are QR codes. And these QR codes you can read using your camera or your mobile phone and they provide more information on the information that's on the slide. For example, these schematics have been drawn by Nick Spencer, and there's a really great piece in Nature which explains how these vaccines work. You can find it by just going after the QR code. So let's go back to the coronavirus. And you see the schematic here on the screen. The coronavirus that is now causing this pandemic has a couple of components. And I've just highlighted two components here. The first component is on the outside, these red spikes sticking out. And you've heard this probably in the news a lot this year, the well-famous spike protein. And we'll go into that a little bit more. On the inside is an RNA payload with these blue and red and green color, the scribbly thing. And this RNA kind of provides a genetic blueprint for the virus to reproduce. So when a coronavirus infection takes place, it uses a spike protein to get into human cells, for example, in the lung. And the way that works is that these spike proteins recognize proteins called receptors on the surface of the cell. And the way you can see this is that these receptors are locks, and the spike proteins on the virus are keys. And the coronavirus that is now wreaking havoc all over the world has a really good key to go into certain cells in the body. So once it's inside of the cell, it releases its viral RNA payload. It comes out of the viral particle, and then two important things happen. The first thing is sort of a copy-paste process begins. That viral RNA that came out of the particle is being replicated, so it's being copy-pasted, so more viruses can be made. The other important thing that happens is that messenger RNAs are being produced from that viral RNA. And these viral messenger RNAs are templates for the viral proteins. Right? So this is the double function of the viral RNA, producing more RNA and producing RNAs that form the template for the viral protein. And of course, when you take these two things together, the viral proteins, such as that spike protein, but also other proteins that make this viral particle, and that copy-pasted viral RNA together make new viruses. And then these viruses are then released by the cell and can infect more cells, and so on and so forth. So that's how the infection takes place. And of course, your body is trying to fight this, right? It is picking up that something is going on, an infection is taking place. So there are certain types of immune cells called antigen-presenting cells. And they are throughout our body, and they recognize when something foreign is invading. It can be a virus, it can be a bacteria. And why are they called antigen-presenting cells? Well, if they take up a virus, such as the coronavirus, they process that, and then on their surface, they start showing other immune cells parts of the virus, called peptides. And these peptides, when they are picked up by other immune cells, they cause the immune response, and they also, and very importantly, cause antigen-specific immune memory. So we have this peptide, it's called the antigen, and your immune system recognizes and builds an immune memory to these specific peptides. So next time you're infected with this virus, your immune system is able to respond very, very fast. So this is very important. This process, both the infection and your ability of your body to build an immune response to it, takes days or even longer. So it's a slow process, and that's why people end up in the hospital. Now, let's go to what you can do about this, or how you can prevent from getting really ill from a coronavirus infection. So the goal of a vaccination is to sort of mimic the process, what I just explained before, but without getting ill. So you want to build, you want to activate your immune system, build a memory, but you don't want to get ill. How do you do that? How have we been doing that for the last 200 years against other diseases? And two ways that are pretty common are protein-based vaccines and inactivated virus vaccines. So an inactivated virus is actually the classical method of vaccination. And what happens is you take the virus that causes disease and you use certain chemical methods or other methods in the lab to make sure that, for example, the payload that is on the inside that causes the virus to replicate and cause disease, that no longer works. So what you then still have is sort of the virus, the shell, the particle, that still causes an immune response and still causes your body to recognize and remember that something happened there, but you don't get sick anymore. The other thing that you can do is to break down that entire virus to its proteins, the protein parts. And for example, you can take the spike protein that is on the coronavirus, produce a lot of it. And when I mean a lot, I mean swimming pools full of spike protein from the coronavirus, and use that to vaccinate people. So there are several vaccines being developed based on proteins and based on inactivated viruses for the current coronavirus, but none of them have been approved yet. So when you inject people with these types of vaccines, they can do the same thing as the coronavirus, which is to evoke an antigen-specific immune response and immune memory. Now, the other vaccines and sort of the newer types of vaccines that have been developed, they work in a little bit of a different way. The very interesting way, and this is why this talk is called From Code to Cure, is that we now have the ability to look at this viral RNA payload. And we can see exactly which code is responsible for which part of the virus. For example, we can know exactly which code is responsible for that spike protein that's on the outside. And this gives, of course, very interesting opportunities. Because if you can take that code and you can transform that into a DNA code, you can, for example, use that in a viral vector. What is a viral vector? A viral vector is let's say, for example, an adenovirus, which is a virus which causes the common cold. You take out all the genetic payload and you put a new payload in, for example, only encoding for this spike protein of the coronavirus. So then you create a benign, non-replicating artificial virus that has a DNA in there, just encoding for the spike protein. So it shouldn't cause disease. The other thing what you can do, and these are the mRNA or the messenger RNA nanoparticles, is that rather than taking DNA, so then you have to go from DNA to RNA to protein, you just take the messenger RNA template from that code for the spike protein and put that in a nanoparticle. In both cases, these things can get injected in your arm, in the muscle, and both of them lead to the production of spike protein mRNA. Or in the case of the viral vector, you go from DNA to RNA. And in the case of an mRNA nanoparticle, you immediately dump the mRNA inside the cell. And of course, this mRNA uses the cell's machinery to produce that spike protein. And that spike protein is then shuffled to the surface of the cells gets recognized again by these antigen-presenting cells who process that and evoke this antigen-specific immune memory. So rather than being a genetic drug, mRNA nanoparticles don't mess around with your DNA. They only contain a template to produce that spike protein that is then used for your body to build an immune response and an immune memory. So now we know how things work. What do these mRNA vaccines consist of? There's now 2 approved, or at least one of them is approved, and one of them is emergency approved, which is the Moderna vaccine and the vaccine by BioNTech and Pfizer. So BioNTech being from Germany. And we kind of have 2 important things here. One is the lipid nanoparticle, or the, let's say the box or the nanobubble surrounding the genetic payload, and we have that mRNA payload. So let's first focus a little bit on the, let's say the protective casing in this vaccine, the lipid nanoparticle. So what does it consist of? Rather than being a viral vector or a weakened virus or an inactivated virus, it actually consists of things that occur naturally in our body, such as phospholipids and cholesterol. So phospholipids are, for example, part of your cell membranes, the outer layer of your cells. And cholesterol is, of course, a molecule that we know well. A biomolecule also plays a role in the cell membrane, where it causes stability. And then we have one more component in this particle, and it's called polyethylene glycol, or short PEG, PEG. And we get back to this a little bit more, because this is a synthetic part. So together, these components make up these particles that engulf the RNA payload. And without this protective casing, let's say, these mRNA vaccines would probably not work on such a grand scale. You probably heard about how unstable mRNA is, and that is the reason why these vaccines have to be kept at such low temperatures. And the lipid nanoparticle definitely helps in protecting this unstable mRNA payload, and also helps in shuffling these mRNA into the cell. So one of the people that played a massive role in development of these lipid nanoparticle technologies, Peter Cullis. Peter Cullis is a professor at the University of British Columbia in Vancouver, Canada. And I had the pleasure of working in his lab for about 3.5 years before I moved to Eindhoven, and I learned a lot about this technology. And also, of course, Peter's collaborators and the companies that he started that made all this happen. The second part of the vaccine is, of course, the mRNA payload, and we discussed this. It provides the genetic blueprint for the spike protein of the coronavirus that causes this, our immune system to build a memory. So next time you get infected by this coronavirus, your immune system recognizes this and quickly acts so you don't get sick. This mRNA code, a lot of groundbreaking work on making that code and how to use it as a therapeutic has been done by Kathleen Carrico and Drew Weissman at UPenn. And the way this code works is explained very well by Bert Hubert. And so if you follow the QR code, he will show you, or you can read exactly what is in the mRNA other than the code for the spike protein. So, do these vaccines work? In short, yes, they work very well. And I can show you here on these 2 graphs. So these 2 graphs show the results from these large trials they did on 10,000s of people to see if the vaccine would work. And what you see on this graph is that there's 2 groups. there's people in blue, green that got the vaccine, and there's people that got a shot in the arm, but it contained nothing more than an aqueous solution, water, buffer. And what you see, these little steps means every time someone gets infected by the coronavirus. And what I'm sure you can appreciate is that in the placebo group, over time, people got infected a lot. People that got the vaccine, hardly at all. So these vaccines in these large clinical trials reach over 90% efficacy. And what that means is that in over 90% of the cases, people were protected from this coronavirus infection. And this is a very high number. So the common flu shot that we get every year, or that people get every year, is only about 70% effective, or maybe less. So this showed that these mRNA vaccines, yeah, they really work. The second sort of remarkable thing about these mRNA vaccines is their speed of development. And the speed of development is also an advantage and has also been a disadvantage. The disadvantage being it being developed so quickly that people are critical about that. How is this possible? We've always been told that vaccine development takes at least 4, 5, 10 years, and now you can do this in a year. I'll quickly take you to through this timeline of what happened in 2020. So already in December 2019, the first messages from Wuhan in China came out about a sort of SARS or SARS-like pneumonia outbreak. And it didn't take long until a Chinese scientist, together with an Australian colleague, published the entire sequence of the novel coronavirus, which they then had identified was causing this pneumonia-like disease. So we're talking a month, maybe less. Because we knew the genetic code, or scientists knew the genetic code, they were able to really quickly act on this and develop an mRNA, let's say, encoding for the proteins of this virus. So in January already, companies like Moderna and BioNTech had already made these particles containing the mRNA. And therefore, they could start testing really quickly. They had done a lot of safety testing and preclinical testing before with similar types of particles. So in March, April, May, the first trials of Moderna and BioNTech in humans started. And these phase 1 and 2 trials are mostly to demonstrate the safety and to determine which dose is needed to get a therapeutic response. July, August 2020, the results of these studies were published in Nature and the New England Journal of Medicine. Turned out to be safe, turned out to be a dose that could be used in further trials, and these large phase 3 trials started in 10,000s of people. Those results were announced in November, was all over the news, 94% efficacy, 95% efficacy in these trials. And maybe a month later in December, the first vaccine was approved by the FDA, or at least emergency approved. And at least in a lot of countries, not the Netherlands, but in a lot of other countries, they started vaccinating people. This fast timeline was only possible because companies like Moderna and BioNTech had already a lot of infrastructure in place to make mRNA therapeutics. This was already coming to the clinic. The pandemic helped in a way to speed up this development because a lot of money became available to do these large trials. And mRNA therapeutics, to go from code to cure. In theory, the CEO of BioNTech said, if we need to change the vaccine, we only need to change the code, and we can produce different types of particles. They're the same box, the same casing, only the genetic payload is different. And we can do that within 6 weeks. So it's really this new technology that enabled this rapid development rather than anything else. Now, how expensive are these vaccines, and how do they compare against the other vaccines that are there? So I've been talking about the Moderna and the Pfizer BioNTech RNA vaccines. Of course, in the news, we've also heard a lot about the AstraZeneca vaccine being developed in combination with the Oxford University. That is a viral vector-based vaccine. And there's also the Sputnik 5 vaccine being developed and approved in Russia, also based on a viral vector vaccine. And you see that because DNA is more stable, these vaccines, these viral vector vaccines, are a little bit easier to produce, easier to keep. Of course, viruses have evolved for millions of years to keep this stuff very, very stable. And for all of these reasons, storage, development, the costs for these types of vaccines are lower than for the RNA-based vaccines. However, if you look what a shot costs, so we're talking maybe around 30 euros, those costs are relatively low compared to the costs that are now related to this, locking down the country for a year. And if I can get my hands on it, I would happily pay this to get vaccinated. Now, the last part that I want to address before we go into the Q&A session is the safety. There are obviously also concerns about, this has been so rapidly developed. Are these vaccines, these mRNA vaccines, are they safe? Now, the most common side effects are also very common for other types of vaccines. So there might be pain at the injection site when you get a shot in your muscle. That is quite normal. You can get a fever, which is usually an indication of your immune system building a response, fatigue, or a muscle ache or a headache. In about 1 to 100,000 people, a rare anaphylaxis or severe allergic reaction occurs. And it looks like that reaction has to do with a hypersensitivity against the polyethylene glycol. And if you remember from a few slides ago, this is this synthetic component. This is on the outside of the particle and prevents these particles in solution from clumping together. And this polyethylene glycol, it sounds really, really scary, but actually, PEG is used a lot, commonly used in food, commonly used in cosmetics, commonly used in medicine. So if you've ever washed your hair or eaten something or taken a pill, you've probably taken up polyethylene glycol. These mRNA vaccines, as I mentioned, because we are working with not a genetic drug, but the precursor for the protein, it's very harmless to the body. All of these components of the lipid nanoparticle can be broken down by the body, and the mRNA is read by the cell a couple of times to produce the spike protein, after which it is broken down. So within a matter of days, the vaccine is out of your body. The immune memory that it builds stays in your body at least for months, hopefully for a lot longer. We don't know yet if this vaccine is something that we would have to take once, or it is something that is more like a flu shot and we would have to take every year. So very little side effects. Now with this, I think I've been talking for about half an hour, so we're coming to the end of my talk. If you are interested in learning more about mRNA vaccines, on the 17th of February, there is a webinar by Katie Whitehead and Yizu Dong, two of the leading scientists on lipid nanoparticle nanotechnology, mRNA and RNA vaccines and RNA therapeutics. So you can find this webinar and maybe get more information there. And I just want to leave you with this cartoon, which is exactly what we're trying to do. We're trying to have cells, eat a piece of genetic code. They express the spike protein, and this is how we launch the immune response and hopefully protect us against this coronavirus. So with that, I come to the end of my talk. I hope it was informative. And if you have any questions for me, we will now move to a Q&A session via Zoom. You can find the link in the description of the live stream. And I'm happy to answer all your questions. And if you don't have time or you're watching this in a later time point, you can also find me via Twitter. So thank you very much for listening.
Hey, you're still here, so you like the video. If you want, subscribe and click the bell.